All right, let's finish this up. So the last video that you would have seen is me going through this on the board, and let's look at all the other possibilities for the ways that the complement system can be activated. So in general, something is going to trigger recognition. This can be something like an antibody, but there are other pathways. There are four pathways, and the classical pathway is going to be the most important and the one that I need to make sure everybody's got. And then the other pathways are just build on this. They're doing the same thing in a slightly different way, essentially. Yeah, and so then we lead to the amplification. So if we get a C3 convertase, we can actually make a whole bunch of activated C3s, which can then serve as attachment points for all the other stuff that we need to make in order to make the membrane attack complex. Then you've got all the little leftover bits that are going to make inflammation happen and call in cells to do phagocytosis. And you will also make that membrane attack complex that will physically punch a hole in whatever membrane is nearby. So there are lots of different factors, and they sort of break down into various groups. You got your C1 factors, and so C1 and the mannose binding lectins. That's what MBL stands for. These are the names of some particular ones. These will also be alternative ways to start off the recognition. So these are each going to bind different things. C1q is going to look for antibodies, and the mannose binding lectins are going to look for mannose sugars, which will be on the surface of some bacterial molecules. There's mannose on your cell surface proteins as well, but we cover up that mannose with a couple of other kinds of sugar so that it cannot be recognized. And that's basically the function of the Golgi apparatus. The Golgi apparatus lets us have mannose binding lectins as a general defense, because if they can bind something, it means it's not yours. Or something's gone terribly wrong, and either way, yeah, it should die. All right, at that point, you have a lot of different activators. So C1R and C1S actually come as part of C1Q over here. They are enzymes that are going to activate the rest of the process. If we're talking about the mannose binding lectins, then that's going to be the MASP proteins, MASP1, 2, and 3 over here. Those are going to bind to the MBL and do effectively the same job. Anyway, what the goal is here is to activate C4. And once you've activated C4, C4 is actually going to stick to the membrane of the bacterium or whatever it is that we're trying to kill. And so that is going to serve to nucleate, like form a center for our complex. C3 is then going to bind to C4. It's going to get activated by one of these things, once again. And from that point, C3 and 4 are going to activate C5. And once you've got C5 activated, C5b is going to call in all these other things. So 5 is going to bind to 6. And 5 and 6 are going to float around. They're not really attached to anything. But 5 and 6 can bind to 7, and 7 will actually just barely anchor into the cell membrane. 5, 6, and 7 will then call in number 8, which anchors very deep into the membrane. It's like like an anchor. And then 5, 6, 7, and 8 will call in a whole bunch of C9s, one after another. And the C9s will make a pore that actually punches a hole in the bacterium and keeps you alive and healthy while killing off whatever is trying to hurt you. Now, there are a bunch of complement receptors. These are actually looking for activated versions of some of these proteins, and so they are the ones that are going to bind all the mediators of inflammation, like C4A, like C5A, and like C3A. And then there are a bunch of regulatory proteins that we could go into if we were doing this for an entire year or if this was med school, but for now, I think this is going to be enough for you to do and for you to learn. All right, so classical pathway, the one in the middle, that's the one that we are going for here. And so here's our C1Q molecule, which is those 5 little threads on the end of it. It's going to bind to 2 different antibodies at the same time. And C1R and C1S are going to be nestled in between its little legs there. And when it binds to 2 antibodies, it stretches out just enough that R and S can activate each other. and then S is going to go on and activate the other things. Or you can say C1 is going to activate the other things, but C1Q is there to regulate R, which is there to regulate S, which is there to go out and do stuff. So it's a system with many outs basically built in, a lot of safeties built in so that we don't accidentally activate anything. So these guys are going to activate first C4, which is going to stick onto the membrane of that cell or bacterium, whatever it is. It's going to call in C2. C1S is also going to activate C2. And so, boom, we've now got double activations of C4 and C2. And so the parts that stay behind are C4B and C2A. Those two together will call in and actually activate C3. And they can activate a whole bunch of copies of C3. So there's a lot of C3 floating around in the blood, and they can just keep popping off the little C3A on the end, that weak inflammatory mediator. And C3B is right there. And then from there it goes on, and eventually we will either make the membrane attack complex, or the C3As will call in some white blood cells, or we'll do some other things that the C3B acts as kind of like a handle so that phagocytes can grab onto these bacteria and pull them in. It's just another way to grab something that that doesn't really belong in your bloodstream or anywhere in the body. Okay, so what's C3 look like? Well, it kind of looks like this. So shown here, these are domains of C3. So just think of them as folded up pieces of the protein. And you got your two chains, and they're going to be linked by a disulfide bond here. This little nubbin right here is going to be the C3A. And so when we get converted, we're actually just going to cut that off. And then all the rest of this can stick together, 1, 2, 3, 4, 5, 6. And the TED domain is going to bind onto the membrane. And that is going to anchor this down so that we can start to build up these complexes. So our first goal is going to be to convert C3 into its active form, which is C3B, which is the B is like the big half usually. In every case except for C2, and I don't know why they named it that way. So usually the A part goes away, think of A away, and the B part is usually big and is going to stay there. There are lots of ways to convert C3, but they are going to be used. So this is where all these different pathways are going to come together. The classical pathway, the one that we talked about, is C4B and 2A together, so C2A and C4B, stuck to each other, both activated, and they are going to activate C3 and turn it into C3B. But there are other ways. You can have one C3 that actually cuts itself, or just breaks for reasons that are quite variable. And then as long as factor B is there, and factor B is this big B, factor B is going to be converted by the C3. It's going to be factor BA, which is going to go away, and factor BB, which is the big part, is going to bind to this. And those are now going to be able to activate more C3 and make more little handles on there. You got your lectin pathway, which is going to use 4B and 2A. It's just going to grab them with a molecule that looks like C1 and works like C1, but is a different named molecule. But it's kind of the same, and it's probably descended from a common ancestor, I would bet. Anyway, once you have got those, the only reason you make C3 is so that you can activate C5. And remember, we could stop at any point up to activating C5 and just have a little bit of a response. But once we go C5, yeah, we're not going back. So here we go. Let's activate C5. The classical pathway, it's going to be 4B, 2A, and 3B, the newly activated C3. They're going to activate it. Same thing in the lectin pathway, because the lectin pathway is basically the classical pathway, but just starting out from sugar binding. And then the alternative pathway, wait a sec for this to clear. There we go. Going to be two molecules of C3B and a factor B molecule. Those together are going to activate C5. That's like the other way, the alternative way to activate C5. And hey, doesn't matter how you activate C5, just matters that you activate it, because when you do, then something is going to die, something bad. So why? Okay, let's talk about the lectin pathway. Lectins are proteins that bind to sugar molecules. And so shown up here, these are mannose sugars, these little green circles down here at the bottom and in here. And in things like a yeast, For example, like a fungus, you're going to have lots of mannose just floating around on the surface. And so if you combine those, you have just recognized something that's from a fungus that is not your own and is going to hurt you. And let's get rid of it. Don't need that fungus in there. In our own cells, we actually cover up those mannoses. They're at the bottom there, but we cover them up very thoroughly so that lectins do not have enough that they can grab onto. All right. Then you have something that looks just like C1, but is actually the mannose-binding lectin. So there's two different versions of this. They're going to bind, instead of C1R and S, they're going to bind the MASPs, which are proteases associated with the mannose-binding lectin, mannose-activated serine protease, I think. And these things are then going to go on and activate the usual factors, so just in the normal way. So the MASP number 2 is going to activate complement 4. We're going to turn it into complement 4B, which is going to anchor in the membrane. And it's also going to activate complement 2, get rid of 2B and keep 2A. And so you make 4B, 2A. And those two together are enough to activate complement 3B and make a whole bunch of them that are then like handles or like sites where you can put that membrane attack complex because they can convert C5. Why do we draw them this way? Well, because they look this way. How's that? Here's the entire C1 molecule with C1Q being the big part. You can see the little 5 prongs down here. And C1R and S being down here. Oh, actually, it looks like there's 6 prongs in those. So, been drawing it wrong. Oh my gosh. Oh yeah, there's 2 around the back. So I guess that was just a perspective thing. So this whole thing is just considered one C1Q molecule, even though it is made of 6 distinct chains that are each trimers, which is 3 copies of the same distinct chain. So that's just, yeah, if you're interested how this stuff actually works. And so classical pathway, we covered this. But you can look in here and this may help. This and those 2 videos and the video that I made, between these things, you're going to watch enough videos to the point where this actually makes sense. That is the goal. All right, but what if you don't want to do things normally? Well, why? The alternative complement activation pathway is for you, sir or madam. So, step 1. If C3b is made, so complement part 3 can sometimes self-activate. Because remember, C3 is actually a protease, so occasionally it can cut itself. Yeah, doesn't often happen, but it can happen occasionally. And there are other factors that can make this more likely, I believe. Anyway, then factor B comes along, which is like the equivalent of like a C2 kind of factor. And anyway, once these 2 are together, then boom, 3 and fake 2 here, which is called B. This is going to get activated. BA is going to go away, and BB, the big part, is going to stay around. And these are then a C3 convertase. So they can make lots of C3s into lots of C3Bs that are all going to be stuck to the membrane right down here. And factor D is going to come along, and that's going to be one of the things that helps do that. So factor D is going to activate factor B, and C3 can sometimes activate itself. That's the C3 activated with H2O. And so yeah, once this gets set off, it is potentially a way to start to make membrane attack complexes or do all the other things that the complement system can do. All right, so once you have the big part of B and the big part of C3, so C3BBB is what they call that, then you can bind factor P to that. So that's factor P, C3BBB. There we go. And that together is going to be the factor P is just a stabilizing protein. And it's going to allow these things. So they have all the enzymatic activity to actually convert C3, but the factor P makes it a little bit more stable, makes it more prolific. There, factor P makes the alternative activation prolific. How nice. Okay, so what are those components again? Yeah, you got your factor B, factor D, and factor P. Together with a C3 that's maybe activated itself or been activated some other way, these guys can assemble. And if factor D activates factor B, which is bound to a C3B, then boom, it's going to chop off BA, which goes away. And BB, it's just tiny BA, so it probably doesn't have any real function. Or maybe, I don't know. I don't even want to speculate on maybe. Yeah, BB is then going to keep C3B active, and factor P is then going to come in and stabilize that whole alternative pathway mess. Okay, so yeah, there are a lot of factors that actually will regulate how much complement is activated. And like I said, until you get up to activated C5, there are ways to turn this down. So, for example, things like decay acceleration factor, DAF, and some of these others, C1 receptor, for example, are ways to basically mute the complement pathway. So, if you started to assemble something but there are no other signs of danger, then there are other proteins that will kind of slowly but eventually come in and shut down that complement pathway. So that, just because a C3b gets activated on its own doesn't mean that necessarily you just start attacking everything, which is very good. So here's a nice chart that's kind of showing all the different parts of all of these and how they relate to each other. So you got your C1s. That's the main activating protease. And remember, that's activated by C1R, which is activated because C1 gets pulled a little bit in different directions by multiple antibody proteins. And it's on those sort of bendy stalks, if you remember that. It kind of looks like an upside down bunch of flowers. For the lectin activated pathway, it's going to be those MASPs, which are just little serine proteases that are going to be activated after mannose binding. And then you also have factor D that can also be a initiating serine protease. And so all these are actually similar looking enzymes, work in similar ways on similar targets. They just have names that would seem to indicate they're quite different, when in reality, not so different. Okay, which component binds to the cell surface? Well, normally it's going to be C4b. That's going to do it. But if C4b doesn't, and we alternatively activate C3, then C3b, which is alternatively activated, that version only, can actually directly covalently bind to the cell surface. And so it'll serve as an anchor point in the alternative pathway. The convertase is then going to be, we're going to bind B, which is going to be activated by D, or we're going to bind C2, which is activated by C1 or by the MASP, C1s or the MASP protein. And together, those are going to activate C3, which is then going to stick with these things and activate C5. And once you've activated C5, you don't go back. There are various cellular factors that can control activation, like a C4 binding protein is what C4BP. It's just going to bind this thing so that it can't actually covalently bond to the cell surface and recruit C2 and C5. So that's a way to turn it off. Yeah. All right. And yeah, you got various factors that'll do local inflammation. The effector pathway, though, that's making the membrane attack complex, that's going to be driven by C5b. And the whole point of all this stuff above is to find some way to make C5b happen. At the right time in the right place, of course. Okay, so this looks like the standard pathway on this side, classical pathway, and the alternative pathway here. And from this point, the classical pathway is the same as the lectin pathway. It was just we had a different activating protease. We had those MASPs that were activating it instead of C1s. So there's our C5 is going to bind, our C5 is going to be cleaved, C5a is going to go away and cause major inflammation. C5b is then going to be free and ready. And these are a bunch of things that can, or rather, these are proteins that can bind to different parts of the complement pathway in various states of activation. And they're on different white blood cells, and they can have different effects. Yeah. Some of these will make phagocytosis happen a little bit more Some will, yeah, they just have different effects on different types of cells. I think that's a good way to think of it. So C3b, remember, can be a handle all on its own. So there are C3b receptors called the CR1 is one of those. And those will actually just bind to C3b and pull it inside of the phagocytic cell, which will then, yeah, actually be under stimulation with C5a. So this will together make the macrophage or neutrophil eat up the bacterium or virus or whatever it is, and then go display little parts of it to the rest of the immune system so we can learn. Okay, so C3, um, yeah, C3 is going to be the C5 convertase. I think that's the important thing here. Let's skip past the rest of that. And let's see, C3A, 5A, and 4A, so the things that we throw away, are all going to leak out into the blood stream. And when they do, there'll be like a constant source of these things coming out. They are then going to call in various cells, various white blood cells of all different types, which can then have other functions. Because generally, you want to sick your entire immune system on a problem, whatever it is. There are a lot of ways that the immune system can solve it. Not all of them work every time. And so your best bet is to just go with a really broad sort of attack. That's a good way to do it. Okay, membrane attack complex, easy. C5 gets activated. 5A goes off and mediates inflammation and makes those phagocytic cells eat more. It's the munchies, basically, for your macrophages in particular. C5b, though, is then going to initiate assembly. So this is like the ancient television show Voltron or something. I don't know. We're gonna clip all these things together, and C5 will form the head, and then C6 comes in and binds to C5. So they're just floating around. C7 is then going to insert in the bilayer. C8 inserts all the way into the lipid bilayer, so it's going to be thoroughly anchored in the membrane. And then it's like they're passing this down. It's like they're going to grab C9s, pass them along, and stick them in the membrane. 1, 2, 3, 4, 5, 6, 7, 8, until they have made a nice, complete membrane pore. And from that point, yeah, the bacteria's guts leak out, and it dies. Here's what those pores look like. Look at those beautiful little pores. So this is looking at them, looking down from the surface. Here we're looking at the side of them. There's a pore right there, but there you can see the C9 proteins actually all lined up, kind of just like that. Yeah, making the little pore. And now the inside is connected to the outside, which means that that cell is about to die. Yeah, so there is naturally a lot of regulation. There are things like protein S that can actually get in there and say, stop the membrane attack complex formation. So we have different ways to try to call this back at different stages. But for the most part, once we've gone past C5, we are already in a state where we are going to have high inflammation. And basically, yeah, some version of an immune response is going to happen. You can just try to slow it down a little bit with things like protein S. And so, at what stage can we regulate this? Well, most every stage, but not quite as much at the end. So, yeah, there are ways to slow down or potentially inhibit the activation all the way along the complement activation cascade. Now, complement is something that is very difficult for bacteria to evade. There are a few molecules that have been developed in just a few bacteria that seem to have some effect, but none of these is completely effective. Complement is actually a really good thing. This is, yeah, going to be eventually effective against pretty much any kind of bacteria that would get inside of you. And so there are ways that bacteria try to fight back, but none of these is quite as efficient as the complement cascade, because it's got that built-in amplification system right there. Once you've converted a C3, it can go on and convert more C3s, more and more and more, and they can then go on and convert a whole pile of C5s, and yeah, there you go, lots of membrane attack. So I posted this in the link. If you try and copy it from this video or the PDF and paste it, I think because there's a line break here, it'll put in a %20, and you have to delete that out. So it's just the D and then the C, DCD52, just like that. That will then get you to the actual video, and that is another excellent video. It's something like 2 minutes, and it does a beautiful job on the classical pathway. And once you got the classical pathway, all of complement is open to you. All of this is yours. So thanks very much. This has been Complement and Chapter 2.